My name is Sierra Cannon, and I will be discussing the colonization of the gut by microbial populations in early life stages of ruminants. Ruminants can be found all over the world and are successful because they have the ability to transform energy from sunlight in the form of plants into energy they can use, as well as products for humans like meat, milk, and wool. The activity of the fungi, bacteria, archaea, and protozoa found within their gastrointestinal microbiome allow them to do this. Microbial colonizers begin to establish within the ruminant from day zero, and their population regulation and change is important to the overall health of the animal. Studying the initial colonization as well as the development of a young ruminant's gastrointestinal tract is vital because it has implications for young ruminant production and health in the present and in the future as an adult. Microbes are acquired through many different modes of transmission. Newborn ruminants are said to have no immunity and are sterile at birth. Yet as early as day zero, they have colonies of microbes already shaping their microbiome. It has been found that most bacterial transmission comes from the vaginal canal as well as the breast milk of the mother, and that the colostrum plays a vital role not only in establishing the immune system, but also the microbiome of the offspring. Fungal and protozoal acquisition is also based on environmental exposure. Fungal acquisition is much easier than that of protozoal transmission because fungal species retain their viability in feces, soil, and feed in the environment, while protozoal transmission must come from direct contact with an adult of this species and is typically transferred through saliva in the water supply. The colonization process proceeds in three major phases, which tend to correlate with three phases of physiological rumen development. But first, I'll discuss the colonization process. The three phases of colonization are the initial colonization stage, which occurs during the first 2 days of life, but mainly at day zero. The transitional stage, which occurs 7 to 21 days, and the mature stage, which occurs 28 to 56 days and onward. At day 56, the microbiome is established and is stable, and not many changes will occur. It also becomes very difficult to manipulate at this time. Studies have confirmed that the microbial populations during each phase were distinct from one another, which tells us that these are different phases of colonization. The figure below illustrates the different periods of colonization and the type of food that is correlated to that time period. And you also kind of start to see the relationship between the rumen development as well as what is going on with the microbes. So these phases of rumen development are simultaneously taking place while the colonization is developing. So the three phases of rumen development are non-rumination phase, which is birth to 21 days, where the young ruminant is only drinking milk, and it is bypassing the rumen and going straight to the abomasum through the esophageal groove. The transitional phase, which is 21 to 56 days, where you start to see the transition into ruminant, and you have kind of weaning in this area as well. And finally, the rumination phase, which is from day 56 and onward. Each of the two processes affect the other's success. As the rumen develops, it affects the bacterial diversity, and the alterations in physiology, function, and metabolism as the rumen wall matures causes shifts in the biodiversity of the microbiome. And the figure below illustrates the physiological changes the rumen wall goes through during these phases. So you can see it's smooth as pre-ruminant, and then you go through the transition and you start to see all the papillae come in as they start to begin to eat more solid feed and roughage. Many studies describe the microbial colonization of the rumen as successional. Colonization begins when facultative anaerobic proteobacteria scavenge oxygen. The pioneer bacterial species create the anaerobic conditions needed for the success of colonization by obligate anaerobes, which increases the complexity of the microbiome. As these successive waves of colonization occur in the developing gastrointestinal tract and microbiome, the microbial populations converge, and this allows the population structure of the microbiome to become more stable. It has also been described as coevolution between the host and the microbiome, because, like I said, as the rumen's developing, they're kind of giving feedback to each other, and you kind of have this coevolution. And you also see the coevolution within the microbiome as well. As the young ruminant develops and moves through the 3 phases of rumen transition, the bacterial population changes in response. For example, a study found that there were major differences in the bacterial populations of 1-day and 3-day-old calves. At each stage of development of a ruminant animal, there are different food sources, physiological functions, and metabolism which all correlate to the diversity of the microbiome, as well as which type of microbe dominates. During the early stages of colonization and development, the microbiome is predominated by the Bacteroides genus, which is replaced with Prevotella by the end of the transitional stage. After day 56, the core microbiome is considered stable and a mature microbiome. In that core microbiome, the main families of bacteria are Bacteroidetes, Firmicutes, and Proteobacteria. So how can we manipulate this to our advantage from a production standpoint? The plasticity of the young ruminant microbiome provides a chance to manipulate it in order to increase the aspects of production and health long term. However, the window is very limited. Possible management practices that producers could use to manipulate the microbial population includes prebiotics and probiotics, dietary interventions, and feed management. Changes to conventional feeding management can positively impact the community of microbes within the rumen. This can look like supplementing with fermentable carbohydrates or beginning to introduce solid food diets pre-weaning, but all of that can positively affect how quickly the microbiome develops and matures and how successful it is. In addition, a study by Palma Hidalgo et al. Looked at the effects that inoculating young goats with different adult rumen fluids had on the rumen microbial development. And from their study, it was found that when these calves were inoculated with the rumen fluid of adult goats fed forage or concentrate-rich diets, the colonization process was accelerated, and that led to an increase in diversity and richness in bacterial, protozoal, and fungal communities. In conclusion, understanding the process of colonization and how it correlates to rumen development and the overall health of the ruminant is imperative. The interactions between microbes and the host are intricate and dynamic and can have cascading consequences, positive or negative, on the success of the host. Through understanding the mechanisms and processes of colonization, producers can manipulate the microbiome in order to achieve peak efficiency and production while maintaining the health of the animal.